Hello, everyone. It is my pleasure today to introduce Dr. Hervé Louche of the Center for ImmunoPHEnomics. Hervé studied developmental immunology in the lab of Bernard and Marie Malison, where he developed a system combining lineage tracing and multi-parametric flow cytometry to elucidate the molecular signaling underlying lineage decisions of immune cells. In 2012, Hervé joined the Center for ImmunoPHEnomics in Marseille, France. With its cutting-edge expertise in mouse genetics and immunology, the Center for ImmunoPHEnomics aims to develop and analyze, in a massively parallel and standardized mode, genetic mouse models to understand the function of the mouse immune system under physiological and pathological conditions. Hervé is the scientific director and R&D manager of the immunophenotyping module at the center, where he drives the development of new phenotyping assays and analysis pipelines, such as a new high-content flow cytometry panel of immune cells at basal, inflammatory, or infectious conditions. He contributes to national and international phenotyping efforts, such as the International Mouse Phenotyping Consortium. Finally, he is leading the mass cytometry effort at the Center for use in functional phenotyping. We are excited to have Dr. Louche present to us today a webinar titled More Parameters, More Informative Phenotypes, Systematic Phenotyping Approaches. Thank you.
Thank you, and hello everyone. So I will present you how we actually work at CIPHE and also how we use the instrument, the ImmunoPHEnomics in that context. So first of all, the aim of the Center of ImmunoPHEnomics where I'm working is to actually understand better the immune system, and so that our substrate of interest is actually highly complex because there are lots of cells that need to interact together to actually fulfill the function of the immune system. So you need to understand and tackle this complexity at first. And we are doing that in the mouse with the idea that we can transfer the information that we will gain on human for better understanding also of the human system. And because of that, it's mostly we actually mostly rely, all of us, on the information that has been published and that is present in the literature. And most of the time, mouse studies are actually being performed by individual labs, and it has been shown that the data that is coming out of these studies sometimes is hard to reproduce because most of the time there is a low statistical power due to not enough replication of the work. So that's why at the Center of ImmunoPHEnomics, we try to approach the problem from a different angle and use systematic approaches to actually be reproducible and be able to compare results between themselves. So I'm working at the Center of ImmunoPHEnomics. You see where it's located. And here, where I'm working in the module of immunophenotyping, the idea is really to blend several things. So one is fancy mouse models with either the disruption of the gene by knockout, but also we could study reporter lines like knock-ins, fluorescent reporters. Combine that with instrumentation of high-end instrumentation, and then blend that also with bioinformatic analysis pipelines to be able to fish out, perform first high-content analysis, and then fish out immunophenotypic signatures that we could correlate to either a new therapeutic agent or the function of a gene or a transduction cascade, and so on and so forth. And for this, what we have at CIPHE is a suite of instruments of different kinds. So we have conventional flow cytometers, like the ImmunoPHEnomics, but also instruments from BD, the Fortessa, Canto. Then we have a spectral cytometer, the SP6800 from Sony, and the mass cytometer. And lately, we also invested in the 10x genomic technology to be able to do the Chromium, to be able to do single-cell genomics. But obviously, and for that, we have defined high-content panels that allow us to profile the whole mouse immune system in different organs and looking at different cell types by conventional flow cytometry. But obviously the star of today is the Attune NXT. So we have been here early adapters. So we started with the legacy instrument that had only 2 lasers, 6 colors. And then we upgraded this to the Attune NXT, again 2 laser, 8 colors. And recently we took the V6 version of the Attune NXT, which is a 3 laser and 12 color instrument. So I'm going to present results taken out of this instrument. So the purpose of what we do is to, with the help of these different panels, is to capture some images in our control mice, and then compare whether these images are present also in mutant mice. And for example, you see that for this mutant mice, we don't capture any difference using these 4 panels. And sometimes we do, with one of the panel, capture one difference, and sometimes with all the panel, we capture differences. And really the idea of immunophenomics is to be able to find what are the cells that are important for this change of image in one of the mutants we are studying. And beside this, it's really important. It was really important for us to also have a pipeline that allow us to analyze lots of samples from lots of mice at the same time to minimize variations. So with this increase of throughput, we actually also combine that with different mouse model, disease model, to actually perturb the immune system and see the response of it. And it can be to some inflammation. So we have some standard assays like peritonitis or cytokine storm or IBD and so on. We can also challenge the mice with tumors. So we have syngeneic tumor models on B6 and BAL-C background. We are also starting that on humanized mice with human tumor cell lines. And we can also challenge the animals with infectious agents. And obviously, when you do these kind of studies and you want to have a comprehensive view of how the immune system is actually coping with this challenge, it's interesting not only to look in blood, but also look at different organs at the same time. So that's what we are aiming. For example, in immuno-oncology pipelines, we might want to study the tumors, but also infiltrating immune cells within this tumor, or the draining lymph node and the cells within this compartment. And for this, so that's here a real-life pipeline that we can perform at CIPHE. You see that we can look at the same time in peripheral organ, for example, the spleen, orientation panel that actually describe all cell types, or have some panels that are more focused to a given cell type, for example, the T-cell memory panel that focus T cells. We can look in blood, different types of cells, and also, for example, in the tumor infiltrating leukocytes, extracted tumor cells, we can look at by mass cytometry, for example, for functional status of T cells extracted from this the tumor environment. And also we can look at the same time on the spectral cytometer the tumor cells and profile them. And obviously for this, and to be able to cope with lots of samples, we had to dimension our pipeline. So we actually have some semi machine to actually extract organs. So that's the one from Miltenyi. If we need to enrich, we also have some Miltenyi tools to do that by magnetic selection. And then we use our Fortesas for acquisition. And we have current limitation to this. It's mostly constrained by the time that this experiment takes. So it's really a group work. So many application specialists are working together to perform these big experiments. It's up to 48 individuals, 5 panels we can run on a single day on these 48 individual samples, and only one mucosal type at the moment, because it takes more time to capture this. So having said this, now, so that it works, you really need to immunophenotype and try to actually remove all the noise from the experiment. So that's what is depicted here in red. we really wish to remove the most of the noise that is coming from the experiment, to actually keep only the green signals. That is the one that is correlated to the mutation we want to study, or that is correlated to the therapeutic treatment we are testing. And for that, it's really important to neutralize variation that are coming from at the operator level, or by automatizing some steps like cell extraction I showed you, also cell counting. At the reagent level, so you obviously need to titer, for example, the antibodies you are using to make sure they always perform in the same manner. Neutralize variation at the instrument level, so this you can do with using a set of beads to ensure the sensitivity is always the same. And that's something that has been long maybe put aside, so the variation at the data analysis level. And this is actually now fixed at CIPHE using automated, supervised automated gating. And here is just pictures to show up this work. So from cell extraction to cell counting. So cell counting is performed on the Attune NXT. To magnetic sorting, acquisition, and automated analysis, all that is done following a standard operating procedure to actually be standardized and be able to compare results later on. And we are looking into actually pipetting solutions for our antibody panels, and also solutions to prepare cocktail of antibodies and store them over time, because we see that still some variation is coming from there, and that we need to neutralize that. So now the Attune NXT, because that's the star of today. How we use it at CIPHE. So first of the thing, I wanted to tell you that we actually did not take that for granted. When we look for a solution, we compare that with different instruments. And in our hands, basically, the Attune, already the legacy, was particularly adapted. Why that? Because it allowed us to, with the help of the auto-sampler, to acquire very fast number of samples and determine the absolute count. So that was point A. And point B is that it was very hard to clog the system, mostly due to the acoustic focusing. So making this solution for us a very robust instrument in the way we wanted to use that. So I talked a little bit also of the auto-sampler. So for us, it's the most robust combination, so the HTS system and the Attune NXT, to actually acquire fast and count samples. We also are really pleased by the fact that this auto-sampler accommodates deep well, and that allows us basically, and we will see that in a minute, to count on a brute extract, so raw extract of cells. So without treatment, we just extract the cell, digest tissues, extract the cells, and directly count from that, or dilution of that. So it allows us to minimize the steps until we get the counts of cells. So volumetric counting, as you've heard, we are really fan of that. So what we do actually is so it needs a little bit of assay setup. And you can look at it by using this plot. So when you do when you draw a gate at the beginning of your acquisition and at the end of your acquisition. Why that is because the Attune NXT drives the sample through a syringe pump. And what you see when you actually push the sample into the machine, you push that with some sheath at the bottom. At the end of the sample, and if you count in this area where the sheath is pushing the sample, you will have some dilution effect, and your counts might be inaccurate. So it's only a matter of actually setting up your assay at the beginning from the sample type you are interested in, and make sure that you are always within the stable. You always have a stable count between your beginning and the end of the acquisition, so that you actually do not underestimate the number of cells you have. But once you have set it up for your particular assay, you can use that rule over and over again. That's what we do at CIPHE. And it's just to show you here that we actually, each time we do a counting experiment, we validate our instrument. We ensure that the instrument counts well. So most of the time, the instrument count well is just to make sure that the fluidics of the instrument is clean for this kind of assay. So that's what we do by using counting beads. the count by bits from Thermo, and they should always give roughly the same count. So we make sure this is right, and if the count is not correct, then we wash the instrument, we clean it, and then we pass the counting bits again. And when we get the good count, most of the time from start it's already good, then we can start counting our cells in our samples. And here is just what we've been defining for our own tissue types, so spleen, thymus, lymph node, how we should dilute the raw extraction of cells so that we get a good count of cells, and within the specs of counting, which is actually described in the white paper of Thermo about absolute counting using the Attune NXT. So it's basically focusing or following this rule. So it should not the count should be at an average speed of 50 to 110 events per microliter. And then it's very handy because you have a statistics embedded within the Attune NXT software. The event per microliter, you can multiply that by your dilution factor, and then you get your the number of cells you have in your sample. So now, how we use the Attune NXT and this counting capability, and why it's so important for. I will talk a little bit about mass cytometry then. Mass cytometry, it's basically the same as flow cytometry at the beginning. But the only change, I would say, is that you stain cells with antibodies that are coupled to metal and not fluorochrome anymore. Then you pass them through this machine, one after another. And then you have a step where you actually put these cells into a plasma of argon at a very high temperature, so 5,500 Kelvin. And the result of that is that each cells are actually transformed into an ion cloud. And then this ion cloud, within this, we will count the number of elements that are present onto the antibody. So I won't go into further detail on that because that's not the aim of today. But one thing that we noticed very early on is that if you pass your sample without counting the number of particles you have in it, and you look at the patterns of expression, for example, here, I'm looking at B cells, so CD19 positive, and I look at class 2 expression versus CD4. And obviously on B cells, I have class 2 expression and no CD4. But what we saw in this early experiment, where actually cellularity was not determined, was that we saw class 2 expression on CD8 T cell. And remember, I'm studying mouse, and that's a real artifact. And we had roughly 16% of artifact in this analysis. And this is coming from the fact that these ion clouds, they need some space in time separating them. And if you pass a sample that is too concentrated in events, what you come to see is a fusion of these ion clouds. So it's similar to doublets that you would see in flow cytometry. So what is key there is really the sample introduction speed and actually the number of events you have per your cellularity. And this can be really, really drastic, leading to really lots of artifacts. So if you actually gate on singlets or doublets and use a different concentration of your sample, you see that over a given threshold, you see that very drastically you increase your percentage of doublets. And for example, if you use 8 times the recommendation, you are over 25% of doublets within your sample, so which is really bad. So that's why we found out that our optimal ratio was 0.5 million per ml, and that we should acquire at 300 events per second. And because of that, what we do now is that each of samples are counted onto the Attune NXT before we actually inject the sample into the mass cytometer to prevent that we go at cell concentration over our threshold, and like this ensure we get very reduced number of artifacts and doublets. Now what we also do on the Attune NXT is define our yield of enrichment when we do magnetic selection. So here it's an example. Before magnetic selection, in that particular example, it's a tumor of a B cell leukemia, so that we injected under the skin of the mice. So what we want to do here is remove the tumor cells and enrich in infiltrating leukocytes. So what we do first, before magnetic depletion, we count the number of cells and their proportion using the ImmunoPHEnomics. And then we do our magnetic, here it's a depletion. We want to remove the B cells, so the cells that are labeled with CD19, coupled to magnetic beads. And we see that after magnetic depletion on the ImmunoPHEnomics, we can score the number of cells that are remaining, and then like this, calculate our ratio of enrichment, our yield of enrichment. So for us, it's particularly handy because we can calculate this enrichment. We can calculate also the number of beads we need to put in face of our accrued extract to actually perform a very good pure enrichment of our population. So this is also very important. And if you do that, then you can start working on TILs and do phenotyping on this tumor infiltrating leukocytes that have been purified because they are devoid of any tumor cells. Now the next point is really to me important, especially for flow core facilities that are shifting to single-cell genomics, for example, or also in others. So I don't know if you've been there. So I've been actually working in a research institute as a postdoc before. So I know both situations, and now I am a director of this core where we also have people coming and sort with us. And most of the time you can see this kind of situation where the scientist is not happy because he's coming with a sample of 500 million cells that are really precious to him. And after the sort, he only got out 30,000 cells, right? And then he blamed the core facility because he thinks the sort was not well set up by the core facility people. And then the core facility people says that most of the time the counts are off and completely wrong, and that's actually what the sorter sees is a lot less of these cells, and that among these cells 80% were dead, and that's because the cell prep was bad, that at the end of it the cell sorter could not do magic, and that very few cells were coming out of this. So we actually, you need a judge piece for that to actually see who is right and who is wrong, and what we do is that we use the ImmunoPHEnomics for that. So when external people come, we make them count the number of cells in the sample that they bring to the facility, and then we also check the viability of that, and we then believe the values that we get out of the ImmunoPHEnomics, and because it's done in the facility, and both core lab and scientists are actually looking at these results, then we have a very better fundamental for the cell sorting, and then it goes very well. Then another situation where it's really important to very well characterize the sample, so that will be more after the cell sorting experiment. We want to make sure that we have a given number of cells within our sample, and this you can do that also with the ImmunoPHEnomics, and this particularly important when you want to use these cells for single-cell genomics. For example, Site-Seq. So here it's a scheme just of this, where you actually are able with this technology to combine antibody coupled to oligonucleotides to fish out proteomic information, and this combined with the sequencing of the transcript. You can now look at both transcriptomic and proteomic at the single cell level. The only issue with this technology is that it's super costly. So I have put here the price that we get in France for the different steps, and basically because of the price and also the technology, where you actually maximum can study 20,000 cells per sample, and this being really expensive, you better be sure that you put in your assay your 20,000 cells, and that they are in good condition, in good healthy condition, because in these methods, if you put too much, it will directly impact the sequencing depth and the number of genes that you will look at. So if you put too much cells, you see less genes, because overall you will distribute the number of reads you have for determining your transcript. So you need to be very precise there, and to optimize also cost. So that's why we did that on the Attune NXT. So we had to adjust a few things. First, the sorting in this given project. We actually used the Attune NXT to be able to sort N of cells to get 20,000 cells per population. So here it was the complicated experiment we wanted to look at. Then there were several steps of optimization. We actually concentrated more the samples, and all that was done actually each time. Each steps were done by counting on the Attune NXT the number of cells we got. Then we could determine the sorting yield also with the Attune NXT to be able to find the best compromise between number of cells, speed of centrifugation, time of cell sorting, to actually ensure the cells were in the top conditions for downstream single-cell genomic assays. That's basically here just depicting the different steps and that we followed, and we found out that 2,400 RPM for centrifugation after sort for 5 minutes let us recover 68% of cells that we initially put after cell sorting. So we went on for this for single-cell genomic assay. So now what I will touch, few words I will touch now on rational panel design particularly on the Attune NXT and the methodology. So I told you our main activity is phenotyping mutant match. In mice, it's actually wrong. We do painting every day, and why that is because we design panels to be optimal for a given instrument to achieve the best resolution of our populations. And we have in the lab 2,500 different antibodies or paint tubes, if we make this analogy. And this comes with different flavors, different colors. So around 30 in flow cytometry and 50 in mass cytometry. And then we have different kinds of pencils, which will be an analogy for the detectors. 18 pencils of different sensitivity, which will be the size of the pencils, basically, both in flow, spectral, and mass cytometry. So when you want to design a panel, you need to take into account all this. And we try to approach that in a rational manner to minimize the time that we spend in designing this panel. So the game is really to find CD marker first that will decipher the population of interest. And once we have that, also to see on which instrument we can run panels or what we want to do. And because of that, we need to know very well our instruments. So first step within panel design is actually instrument characterization step. Then what you need to do, because you want to match the brightness of a dye with level of expression of your molecule of interest for your given panel. But that means you need an a priori knowledge about what is bright and not on your instrument. And also, very importantly, what came recently is that you need to also to look at the spectral overlap of your dyes and the spread that is also resulting from the spectral overlap and spread data. And this is also something that you will see depending on your instrument optical configuration. So you need at first to define that on your instrument. Once you have that, you also need to know something about your biological sample. What are your population of interest? What is the density of expression of this antigen? And whether the antigen you want to look at are co-expressed or not on a given cell type. So again, first step is instrument characterization. You have to assay for the sensibility of your instrument and find out the right voltages to use for your detectors. And we've been developing, Pierre Grenot essentially in the lab has been developing a stepwise evaluation of instrument performances, and I will talk about it today because we did that on the Attune NXT. And actually, this pipeline is completely inspired by the paper of Stephen Perfetto that I just put here on that slide, and I really encourage you to read. It's really a mind for this kind of work. So we just followed that, and we actually characterized several instruments also based on these pipelines. So first of the thing, you need to know the optical configuration of your instrument. So here we are on an Untuned NXT of version 6. So you have tables that tells you what kind of fluorochromes you can look at, the laser power, and so on. Then the next step you need to do is to define a voltage for which you will be over electronic noise. And before, it used to be done using eyeballing. And actually, this is not so good by putting your cells actually in a given on your logarithmic scale on given position, and this should be stopped, because the more colors you have, the more problem this method is bringing to you. So the only idea about that you should follow is try to put your cells over electronic noise of your system for a given detector. And then also never acquire signals that are over the maximum linearity of your detectors. So when you characterize your instrument, you need to find ways to actually look at electronic noise and define what is this maximum of linearity and work within this interval. So the way we do ensure whether we are within this good interval for measurement on each detector is that we use rainbow beads. And on these rainbow beads, we look at different statistics on different peaks. So we take the RCV of the 4 peaks of the rainbow beads, and then we look at the MFI of peak 5 and peak 3. And for this, we determine the linearity range, so which is calculated using this formula. So MFI of peak 5 minus MFI of peak 3 divided by the MFI of peak 3. So that gives you values that will show whether you are linear or not across that range of fluorescence. And then the linearity max is actually being looked at by looking peak 7 and see when the signal is stable. So for example, and whether it's stacked, when the signal is increased. And when it's actually stacked, that means that you reach the maximum of linearity. And then we run these beads across different gains, gains for APDs or volts for PMTs, with a so-called voltration. And then we see the evolution of these statistics. So you see that, for example, the RCV, it's very high. And then when your voltage is high enough, then it's stable and remains flat. And that's actually when you are over electronic noise. Now, when your linearity range so the blue line is stable and then start to fall, that means that one of your peak is actually out of the measurement scale. So, and that's actually you push too much your voltage. And then you see also that linearity max is increasing upon voltage increase, and then you reach a plateau, and that's when you actually your peak is not resolved anymore, so you reach your maximum of linearity. So the window of work that we can work on the given detector here is boxed with red dots. So that's how you actually define what you can use on the voltage range, on the range of interest you can use for your given detector. So by this we run it throughout on all of our detectors on the Untuned NXT, and we define the good interval for work, and we define this minimal voltage for which we are over electronic noise. Then what we do, we try to define what is bright or not, right? So for this is the work of Theo Lustou in the lab, and then it's always the question of what is bright and dim, and this we see that for different dyes that you could detect in a given detectors, we see that the brightness can be very different depending on the dyes. So it's very important to actually check on that, yeah, but you need some a priori knowledge of the dyes, and you never know about the instrument. So that's why you need to do that, and once on your instrument to characterize it. And for this, we've been, but it can be very labor intensive, yeah, to define this brightness and fit it with with the molecule you want to look at. So for this, we try to standardize a bit and do one very big experiment. That's how we establish our brightness index of different dyes. We take out splenocytes, 2 million, and we put them 2 million cells per well, and then we stain them with a non-time mouse CD8, all time the same clone. The only thing that will change, and the same quantity, 0.4 microgram, the only thing that will change is the fluorochrome to which this antibody is conjugated. Then we do our staining in the same staining conditions, so 100 in 100 microliters for all the conjugates. So on the Attune NXT, it's the 35 conjugates we've been testing and that we are fitting our optical configuration. And then upon staining, we wash, and then we acquire on the Attune NXT all the single stain samples. Right. The other thing is that we acquire that at 3 different voltage. So the base, the minimal voltage, the base PMT voltage is actually the one that we defined using the RCV method to be over electronic noise. And then we increase by 50 or 100 volts. It's just this to see that whether on our cells this minimal voltage is actually the optimal one for the resolution with our dyes. Yeah, then we select the different dyes that fits on our cytometer, and this I mentioned that already. Then to define what is bright or not, we use stain index. So here I've plotted the formula of it, and then you can actually derive this bright index. So here is just showing you the values that we got for the basal, and then we look whether we see an increase at 50 volt or plus 100 volt. And we see that most of the time the basal voltage is good, but for example for BV510, we saw that we had to increase by 100 volts to find the best separation between negative and positives. So you need to do that. You can do that with a raw value, or to ease a bit your comparison with normalized stain index, where you actually take the brightest or the higher value as 100%, and then you compute everything towards this. But that helps you to find what your optimal PMT voltage is. And you see that, for example, most of it was the minimal voltage, but sometimes we had to change it for given detectors. And once you have done that, you run again your bright index with the optimal voltage, and you can define this bright index. And yeah, so from that, on the Attune NXT, we could see that BV421 was the brightest dye in our hands, but that all others, I mean, most of them were super bright dyes, so super brights are really super brights. And basically, this helps you don't really look at the relative brightness to each other, but what you need to do is then define categories of very bright dyes, dim dyes, and really bad dyes to actually then later on match with your antibodies. Then the next thing is to look at the spread data, so which is coming out from the spectral contamination between dyes. So we know that dyes, fluorescent dyes, they do overlap. Especially the peak of emission are never fine, so there is some light that is coming from one detector that is detected actually that comes from one fluorochrome that is can be detected in adjacent detectors, and this we solve by compensation and distortion. And the more spectral overlap you have, the more spread data this will lead to. So, and this spread data can actually decrease the resolution of your population. So you need to calculate that. And then here, just an important thing is that the spectral overlap is actually independent of compensation. If you keep voltage, because this is really a property of the dye, if you keep a voltage content for the detector in which you will actually compute the spread, and then you vary only the voltage, the primary voltage on which your conjugate is the best detected, you see that you can decrease or increase the voltage. The compensation will change, but the spread data that we see is always the same. So the game is really to to compute that first, to be able then to minimize the spread data that will be engineered and export statistics. So we use that by using this matrix here that is depicted. So you take the spread value that you compute in the secondary detector, which is here, for example, P size 7, and we divide that by the fluorescent intensity, which is actually given by the separation between your positive population, the MFI of your positive population, minus the MFI of your negative population. And that's really important to do that step, because that will have strong implication for later data analysis and especially advanced methodologies. Here I have actually it's only looked at a single stain color, BB700, that I read on this instrument here in the channel P-Sci 5.5, and then I read unsupervised analysis, t-SNE dimension reduction, and I actually did two of these analyses. One, I omitted the BB700 data for clustering, and one I took it. And otherwise, you see the run input parameter are the same between the two t-SNE. It's only that, but the image is different. On one side, I see two populations. On the other side, I see three with some kind of vival home that we can see, right? So, and that's only because I omitted to take BB700 in this, and which is, and you can see that actually one of the side of this horn is actually the positive spread of the dye, and the other one is the lower spread of the dye. So you really need to be careful, minimize the spread at all costs, because if you then in later downstream analysis, advanced analysis, if you don't use this marker in your clustering, you might create very big artifacts. So and then you also need to set well your transformation around the zero, and actually the more spread data you have, the more your negative will be impacted as well in the multidimensional space. So that's why you also need to minimize this, because otherwise, again, that would have impact on clustering methods, for example, later on. So you need to do that. Actually, it's not something we discovered. It has been published by Enrico Lugli and Mario Roederer recently. So that's actually using the bright index. You have the data to actually compute the spillover spreading matrix with the help of the formula I gave you before, and that's the spreading spillover matrix that we actually define on the Attune NXT. So now we know where these red dots, for example, it shows where it spreads really badly from one detector to another one. So we know what are the worst combination that we should not pick for our panel designs. And then based on that, now I can look for dyes that will actually, I will be able to put on antibodies to define these cell types. Now I'm matching the brightness, because I have the bright index on my instrument. I can bright that to the antigen density, and yes, this is still based on a priori knowledge of the scientist, but soon, at least for the mouse, the data will be released and will be available because we are currently determining the antigen density of all the molecules for which BD and BioLegends have surface have antibodies, and then we can now look from this quantity, see what is high expressed or not, and then match the dyes that we will use for the discovering this population. I have to say that very soon there will be an interface to do these laborious steps of instrument characterization, bright index, and spillover spreading matrix computing that will be available on our GitHub. So I encourage you to look at that. It's to help you go faster to that step for your panel design on your given instrument. Now, what we do, we want to do high content panels. Why that? Because obviously we don't only count on the Attune NXT at CIPHE. We do also some nice experiments. For example, phenotyping of peripheral blood cells, we can do that on the AttuneNXT with a very small volume of blood, and take advantage of the deep well to actually have good resolution of this staining procedure in total blood. So I've put it here after lysis. Here what we wanted to do is transfer one assay, phenotyping assay, on the AttuneNXT. So it was a 10 color panel, reference panel that we had, and we wanted to transfer that to the AttuneNXT, use super bright dyes because we saw they were very bright on our instrument and could yield better resolution, and also integrate new markers for a better deciphering the functional subset of T cells. And with our bright index, the spread matrix, we defined three different panel candidates, and we could see here that, for example, the impact of dye brightness on markers. So definitely you can see that some panels are better than others, and we basically selected the panel-free configuration. So here's just the end of it. I showed you the different rational steps for high content panels that comes from actually a serious instrument evaluation first of the sensitivity of your instrument and the detectors. And once you have that, you can design very quickly high content panels on your instrument. So as a conclusion slide, I can testify that the AttuneNXT is very well built instrument. Otherwise, we would have shifted from since now because we have been using the legacy and all these updates. So we are really happy with the AttuneNXT. For us, the automated auto sampler is a real plus when it is combined with acoustic focusing, because it allows you to count very diluted sample really fast. It's very precise and gives really reliable event counting. We have not seen any carryover because each time you actually inject a sample, the sample probe from the HES system is actually being washed and then is preparing the next sample. So that's really good. That's why I think we see no carryover. It's very robust and resistant to clogging. If you get a clog, it's because something went really bad with your sample, I guess. Which is some instruments are more susceptible to this, but the AttuneNXT is fine. Then once you have defined your optimal gains, it's really easy to use instrument for multicolor applications, and hopefully we will soon release this tool that will help you to do that, also very fast and in an efficient manner. And then I hope also I will have convinced you that when you follow these simple guidelines, you can define high content panels very, very efficiently and very quickly. So the only drawback I see is actually coming from the encoding of the data on the AttuneNXT, the number of bits we have. It might be sometimes tough to actually scale the data around the zero to visualize it perfectly. But I know this is actually something that is undertaken right now by the software developer at Thermo to address that point. So with this, I would like to thank everyone that were involved in this work in the lab, and especially Pierre Grenot and Theo Lusto, who did so Pierre the instrument characterization work, and Theo Lusto the bright index and the spread matrix calculation, and then all other people of the team. Thank you very much for your attention, and I'm open to questions now.
Thank you, Hervé, for that presentation. So some questions that we have gotten are, can you please describe some of your sample preparation of different tissues? So in general, are there differences in how you prepare different mouse tissues? Are there any tips that you have to offer about the best practices for doing so?
Yeah, so there are differences, and actually you need to optimize your cell extraction conditions per organ. So that's very clear. Some tips I should give is actually also to know what the markers you are interested in and check these whether they will stay on the cell upon your cell extraction conditions. For example, you can use proteases that chunk off all the cell surface molecules. You will have lots of cells at a good viability, but then you will strip off the markers you are interested in for use for phenotyping. So you need to check that. One way to do that is actually to mix a sample you know very well within your cell extraction conditions. For example, we usually do that with splenocytes, and then we checked whether during the time of digestion and mechanical dissociation with our other tissue type, whether our splenocytes that we will first label with a given color are found well, and then whether the surface molecules that are on them are conserved or not. So that's one way to check whether your extraction conditions actually don't trim your surface molecule of interest. Otherwise, look at viability, and that's something you can do with the AttuneNXT very, very quickly. So and then, sorry, I did not say that. Obviously, you would need to change to play on the enzymes you use, the time you apply them, and also the temperature.
Thank you. So another question is for the use of for doing cell counts for single cell genomics. It's important to have correct accuracy. Why is it so important that the accuracy of your cell counts come out correctly in that procedure?
It's because if you put too much cells, you will get out very few genes per cell. That you will be able to detect very few genes per cell. So these experiments are very expensive, and if you just run them because you put 3 times more cells than what you actually can on this assay, then it's going to be very bitter because you will have lost time because it's time-consuming pipelines. And money, and a lot of money. So you really need to account accurately. And I can tell you, around us, we started to count on the AttuneNXT these samples, and then because we actually at that time did not provide a single cell genomic part, so it was done remotely at another site. And then people started to use the counts that were given by the Attune because that was leading better results at the end after sequencing of their encapsulated cells. So for us, it's the best way to do this.
So when looking at the spillover spread matrix, frequently people will do this with FlowJo before, but you showed it using Tableau. So why did you choose Tableau, and are there other options besides FlowJo and Tableau for this?
So actually, FlowJo is fine. I mean, you can do it with it. What we like with Tableau, it's only for the visualization, so it's not for the data analysis that we use that. So basically, we read tables in Tableau, and we like using Tableau because we can play with the visualization, we can tune it and put several information. For example, in the spread matrix you saw on Tableau, we can dimension the size of the circles by, for example, the stain index. And then on one visualization, we have 2 information. We have stain index and spread, which is 2 components you need to take into account when you make your choice for picking the right dye for the right antibody.
So just a general question. For a researcher who's just starting out using flow cytometry for phenotyping, in your experience, what have you found to be the most common mistakes that people will make when deciding on a panel and how to run a panel?
Well, most of the time, these people don't characterize your instrument, so that's point A. Sometimes they take what is in the fridge, what antibodies are already available. Sometimes they use a panel that works well on one instrument and thinks it will work well on the next one. I think all that is very common mistake that are being done, and I'm a big believer of the fact that you need to design your panel for your instrument, and that's the way you achieve the best results, because best resolution. And if you follow that, and that's what we do on our several instruments, and hopefully this will be fastened by the application we will provide soon on our GitHub, then, you know, there is really no reason why you should not rationally design your panels for a given instrument. And at the end of the day, you save time, and the data is of better quality, so why not do that?
That's the end of our time for question and answers today. I'd like to thank Dr. Hervé Louche for his time and giving us this presentation. So today's presentation was sponsored by Thermo Fisher Scientific in partnership with Wiley's Current Protocols. This webinar will be available for on-demand viewing at the Current Protocols website at currentprotocols.com. Thank you for joining us today.